Okay, so we left off with the endomembrane system and talking about lysosomes. And so, just quick little review. A polypeptide is made via the ribosome on a rough ER. It's made, buds off of the rough ER, goes through a transport vesicle to the Golgi apparatus. The Golgi apparatus modifies it. It buds off the Golgi apparatus in another transport vesicle and can be secreted from the cell. Hence, the rough ER is known to make secretory proteins, proteins that will be secreted from the cell. Second thing is sometimes those proteins that are made are going to be the enzymes in a lysosome, so that's how the lysosomes are created in the cell. And remember, those are involved in digestion for unicellular organisms and also for getting rid of dead organelles. So we're going to continue on with the last part of the endomembrane system, and that's with vacuoles. Vacuoles look awful lot like vesicles, but they have different functions. So they're large vesicles derived from the ER and the Golgi apparatus. So this is the third thing that can come off, is this is how vacuoles are made. They perform different functions in different kinds of cells, so therefore there are different vacuoles. I already talked about one, that's the food vacuoles formed by phagocytosis that I drew and talked about in the last video on the previous page here, how this is a food vacuole. So this is made, and so it contains food. And remember, the lysosome can fuse with it and digest that food. And so this is how unicellular organisms digest a lot of their food. Contractile vacuoles, found in many freshwater protists, pump excess water out of the cell. So therefore, a contractile vacuole, if paramecium have these, and so here's your little one unicellular organism, and what happens is if it's put into pure water, water can diffuse into it, and if it's not careful, it can balloon up and have a possibility of exploding. And so what happens is these guys have these contractile vacuoles that water that's diffusing in goes into, then kind of goes up against the membrane and contracts. It pushes in and squeezes all that water out, and so therefore protecting the paramecium. And so that they call that a contractile vacuole because of the word contracted that. And then central vacuoles are in plants, and this is takes up a good portion of many plant cells, going back to this picture of a plant cell. This is the central vacuole, and actually oftentimes it takes even more space. And it holds organic compounds, organic pigments, poisonous compounds, water, and so on. So a big component is water, but pigments, like in the petals of flowers, is where those pigments would be found. Poisonous compounds, so organisms that are plants that are poisonous would store those compounds there. All right, and so that's the central vacuole, very large part of the plant. So therefore, we have all of these organelles as part of our endomembrane system. And before I actually forget here, the contractile and the central vacuole, since they're part of the endomembrane system, let's add them to this picture over here. That they actually, let's use orange here. So also coming off of there, budding off of there, could be vacuoles. So that's the third. So 2 was this one here, 3. All right, so there's 3 things that can kind of bud off of that endomembrane system. And so collectively we have the nucleus connected to the rough ER with the ribosomes on it, which is also connected to the smooth ER, so therefore all of the same kind of membranes. Transport vesicle buds off with the finished polypeptide, goes to the Golgi apparatus and fuses with it, and it goes through there and gets modified and buds off and fuses with the plasma membrane. Hence, all of these things are part of the endomembrane system. A couple of other organelles that are important for cells are the mitochondria and the chloroplast. Which one is found in both plant and animal cells? If you answered the mitochondria, you would be correct. The chloroplasts are only found in plants. So the mitochondria are the site of cellular respiration. Metabolic process that uses oxygen to generate ATP. So it uses oxygen, I'm going to put here, and glucose. And we break it down to get energy to make a molecule called ATP. And then chloroplasts found in plants and also algae are the site of, I bet you know the answer to this, photosynthesis. Photosynthesis is when the plants make their own food. This is how you get glucose onto the earth, is by photosynthesis. Now, the thing about mitochondria and chloroplasts is that they're interesting in that they have similarities to much simpler cells. They have similarities with bacteria, which, remember, we talked about is a prokaryotic cell. And so the reasons why they are similar is that they both have a double membrane. So both the mitochondria and chloroplast have a double membrane around it, so two membranes. So do the bacteria. They contain free ribosomes and circular DNA molecules. So does the bacteria. They grow and reproduce somewhat independently in cells. So do, the bacteria don't live in cells, but the bacteria are free living and they can grow and reproduce on their own. And mitochondria and chloroplasts also grow and reproduce on their own inside of cells. So this led to Lynn Margulis, a scientist that came up with this Endosymbiont Theory based upon what she knew about mitochondria, chloroplasts, and comparing it to bacteria. So this theory suggests that an early ancestor of eukaryotes engulfed, means took them in, kind of like the unicellular organism takes in the food that it eats. So engulfed an oxygen-using non-photosynthetic prokaryotic cell. So oxygen-using, that's like a mitochondria uses oxygen in the process of cellular respiration and not photosynthetic prokaryotic cells. And then it also engulfed another one that was the chloroplast. But let's look at this one first. So what do I mean by engulfed? Let's draw this. So here is, okay, so here's your mitochondria, here's your cell. And so what they're saying here is this was a unicellular organism, this is a unicellular organism. And so what this cell did is engulf, kind of go around, around the mitochondria like this and dent their membrane like this, and then you have the little mitochondria inside until eventually you have a cell with the mitochondria. All right? And so, well, in this case, it was a unicellular organism. Now, once it was inside the cell, it became useful because it does cellular respiration, uses oxygen, and so on, and it became useful. And so her theory is that this is how the mitochondria actually came to be, an organelle in a eukaryotic cell. So this is kind of an explanation to bridge the gap between prokaryotic and eukaryotic cells. Prokaryotic cells are the oldest cells found on Earth, and according to the fossil record. Then came eukaryotic cells. So this kind of shows how you might have gone from prokaryotic to eukaryotic cells. So the engulfed cell formed a relationship with the host cell, becoming an endosymbiont. So it was inside, and it did something for the cell. So the cell does something for it. It's a nice little house, and so on, and it performs a function doing cellular respiration for the cell itself. The endosymbionts evolved into and became what we know as the mitochondria in the eukaryotic cells. So then, that's the mitochondria. To explain the chloroplast, her theory was that at least one of these cells that took up the mitochondria and have the mitochondria now, took up a photosynthetic prokaryote in a very similar manner, which then evolved into a chloroplast. So, so they took the chloroplast, or the chloroplast was once free living as well. So there's your mitochondria, there's the chloroplast, and that was taken in the same way. And now you have some cells, they have the mitochondria and now the chloroplast, and therefore have both of those organelles that do something useful for it. So this picture is showing you here engulfing what will be the mitochondria and engulfing what would be the chloroplast. And so that is an explanation. They call that the Endosymbiont Theory because mitochondria are unique and chloroplasts are unique in that sharing all those characteristics with bacteria. So let's look at the mitochondria and specifically what does it do. So they're in nearly all eukaryotic cells. They have a smooth outer membrane and an inner membrane folded into, the folds are called cristae. So what does that mean? So let's draw here. Your mitochondria is kind of bean-shaped. So that's your outer membrane. And then your inner membrane's right inside of that, but it's folded. And so, so we have our outer and our inner membrane. The folds are called cristae. So what this does is create two compartments. So it says the inner membrane creates, and it's two compartments. Where are my two compartments? My two compartments are one right here, and this is called the intermembrane space. And my second compartment's here, and we call that the mitochondrial matrix. And so the matrix is just the space inside of there. And so we can see here that the inner membrane, the folds, what is that? What do you think? Why is that helpful, the folds? What does it increase that we've talked about? It increases surface area. So guess what? The process of cellular respiration, in order to have more cellular respiration occur, it involves this membrane here. So some metabolic steps of cellular respiration are catalyzed in the mitochondrial matrix. So some steps occur right here in this space. And then the cristae present a large surface area for enzymes that synthesize ATP. Synthesizing ATP is the purpose of what happens in the mitochondria, which is cellular respiration. So cellular respiration is to get energy in the form of ATP, and so the enzymes are actually found there. So I'm going to go ahead and put them in the membrane here. So you have enzymes, and so you can see the more surface area you have, the more enzymes you have, which means the more ATP you can make and the more energy the cell can have. And so all of these right here are enzymes to make ATP. And so, that structure of the mitochondria helps a lot with its function, which is to make ATP, synthesize ATP because of the folded membrane and all the enzymes to do that, to speed that reaction up. So when we look at this picture, this is just showing you the mitochondria in a three-dimensional form. So I want to show you here the different parts. So they're showing you here the cell in blowing up and enlarging the mitochondria as part of that cell. This is an actual picture on the outside of the mitochondria, and this is another picture where they dyed the mitochondria and the DNA inside. And so let me show you that picture here. So here's the mitochondria, and the DNA inside the mitochondria is in yellow. All right, this is the nucleus inside the cell, so this is the nuclear DNA, and then you have your mitochondrial DNA in yellow. All right, so let's draw here. You have the matrix, and you have the folds, the cristae. So you have your inner membrane in your cristae as part of the inner membrane. So you're seeing here some of these folds like this. All right, so that's what we're seeing there. And so that's your folds. You have your inner membrane. You have on the outside, you have your outer membrane. So this is kind of showing you a cross section of it here. So this would be like this, kind of looking at, and this is the cross section. And this would be on the outside, kind of, that would be your outer membrane, so you can kind of see that. And then you have the matrix, which is the fluid, the space inside, and you have your DNA in there. So the DNA, it's kind of hard to see the DNA, but let's just put here little circles. All right, that's your DNA, little circles. And then you also have your free ribosomes, which are just little dots. So the little dots here in here are the free ribosomes. And so that is your mitochondria, all right, site of cellular respiration. So your chloroplasts are the site of photosynthesis, we said, right? So the chloroplasts contain the green pigment. Do you remember what the green pigment is called? Chlorophyll. So chlorophyll, as well as enzymes and other molecules that function as the key light-trapping molecules in photosynthesis. Photosynthesis, you need sunlight or light, and so you have to have pigments. Pigments are molecules that trap the light, and we use the light is used in photosynthesis to make glucose. So chloroplasts are found where? In green plants and some algae. And so they're found in leaves and other green organs of plants and in algae. So when we look at the structure here of a chloroplast, we see that this one also has an inner and outer membrane. So you have right here the inner membrane. This is the cross section, inner membrane. And then you have your outer membrane, that's this part here, so you can see the cross section. This is going to be on the very outside part of it. Like that. So you have your outer membrane. But notice that they're not folded. Neither one of them are folded. They are smooth. And so what that tells me is that there's not a lot of surface area, which means that those two membranes probably aren't involved in photosynthesis. But what are there a lot of in here? There are a lot of these little discs called thylakoids. And so the thylakoids, they look like kind of like pancakes stacked on top of each other, or poker pieces stacked on top of each other, but they cut open here, so they're actually open, these thylakoids. And what they look like, let's draw over here. So so each one of these is called a thylakoid. So one of these right here is a thylakoid. And then the whole stack is called a granum. And that is the singular. You will also see it also written as grana. I want you to know that that's the same thing, and that so it doesn't confuse you. That's the plural form. So if you're talking about multiple stacks, we're talking about grana versus granum. And so then what happens is you have the space inside of it. That is called the thylakoid space. So inside thylakoid space. It's really original, but makes it easy. Alrighty, and then they're surrounded by a membrane. So let's write a few things here. So chloroplast structure includes, so the big thing is the thylakoids. So membranous sacs, I just said that they're surrounded by a membrane, stacked up to form a granum. So granum is the stack there. And that's floating around in the fluid inside the chloroplast, and that is the stroma. So inside the chloroplast, around all of this, though it's taking up the space that's not a thylakoid, is the stroma. So that's the fluid, and it's hard to draw that in there. It's just part of the the inner parts of that. And then also remember that the chloroplast has DNA, so put a little squiggly line here, and then there's DNA in there, and that's why it's thought to have once been a free living cell all on its own. All right, so that's the chloroplast, and so the thylakoid the thylakoids. There's so many of them. There's a lot of surface area for thylakoids, and the reason why is because they're so helpful in photosynthesis. And why they're helpful is that there are pigments embedded in the membranes of the thylakoid, and guess what these pigments do? What do? They absorb light needed for photosynthesis. So guess what? The more surface area you have, the more thylakoids you have, the more pigments you have, the more light can be absorbed, the more energy you have to do photosynthesis. And so therefore, huge help in the process of photosynthesis. Alrighty, we're on our home stretch here, and this is with what's called some other organelles that are important, and one of those is called the cytoskeleton. And whenever you see cyto, it's referring to cell. So it's like the skeleton of the cell. It's a network of fibers extending throughout the cytoplasm and help give shape and structure to the cell. So let me explain here. So here's your cell. All right, your cell has the nucleus in it and the organelles and so on. What the cytoskeleton does is have 3 different types of molecules that make it up. Microtubules. Micro means small. Tubules, tubes. So what happens is to help give shape and structure, there these microscopic little tubes that go throughout the cell like this. Oh, my light gave out again. And there are also microfilaments. These are like small, like threads. They're fiber-like. And so I'll do this in red. So therefore, you have them like this going throughout the cell. Like that. Okay, and you have intermediate filaments that also are, they call them intermediate filaments because they're in between sizes. So tubules are a little bit bigger, they're thicker than the microfilaments, which are thinner, and so these are intermediate sizes, and they also go throughout. They're thread-like, just thicker threads that go throughout. So you can see it's kind of like a scaffolding system inside the cell, and so what it does is help keep that shape of the cell. You have to imagine that this is two-dimensional. The cell is like a globe, and so those things are going all throughout and putting pressure on the outside of the membrane, giving it shape and support and structure. All right, so so that's the big thing. And then here, the second bullet says it organizes the cell structures and activities and anchors many organelles. And so down here, I just wanted to show you, this is a microtubule. So I'll put it in the same, I guess I should have put microtubule here in green, color-coded, and the microfilaments in red for that cell over here. So this is a microtubule. So like this, and so it's one of these guys here. They found that organelles actually have these little receptor proteins that kind of ride the microtubules, kind of like the rails on a subway system. So therefore, like the vesicles, remember back in the endomembrane system when we say the vesicles, the transport vesicle goes to the membrane and fuses with it to secrete the proteins. Well, how does it get there? Well, guess what? It has this little motor protein, and it uses some energy, and it glides along that microtubule to the cell membrane and then can fuse with it and secrete those proteins. So it's kind of like a little, subway system inside the cell. It's pretty cool. So this is a picture of that showing you, this is the actual microtubule right here. And these are the vesicles right there, all right, riding on the microtubule. Okay, so that's the cytoskeleton. Another thing that's found in animal cells is centrosomes and centrioles. In animal cells, microtubules, so those are those small tubes, grow out from a centrosome near the nucleus, and they're involved in cell division. They're involved in making the spindle fibers when the cell divides. In animal cells, the centrosome has a pair of what are called centrioles. We'll look at a picture here in a second. Each with 9 triplets of microtubules arranged in a ring. So they're just made out of microtubules as well. You don't have to memorize that it's 9 triplets and so on. So they're in groups of 3, the triplets. So when you look at this picture right here, here's the microtubule, all right, so one of these microtubules, and they're arranged in a ring here. That forms a centriole. Now, how that relates to the centrosome, it's kind of hard to see here because it didn't copy very well. The centrosome is like a region right here, the whole thing is the centrosome, and there are 2 centrioles in the centrosome. And so what happens is, I'm going to try and see if you recall this from biology. If this is your cell, when cell division happens, you get your chromosomes. Do you remember your chromosomes kind of look like little X's? They line up down the center of the cell. And if you recall, you have microtubules that attach to them. So what happens is you have these centrosomes with the centrioles at either side of the cell, and those microtubules elongate and from there and connect to your chromosomes. Do you guys remember this at all? And remember, they're involved in separating the chromosomes, all right, when during cell division. And so that's what the centrosomes are, and those are only found in animal cells. Other things that are found only in certain kinds of cells are these things called cilia and flagella. Microtubules control the beating of flagella and cilia. And let's talk about the difference between them. They both aid in movement, and they're both projections from cells. So the flagella, like bacteria, or this is a picture of the human sperm right here. Have flagella, so they're long and tail-like. Bacteria can have more than one, so depending upon the bacteria, there might be several of them. The human sperm only has one, and it helps for the organism to move. And then cilia is more hair-like little structures. They're smaller around the whole thing. And so this would aid in movement. So the direction of the organism's movement as these guys all kind of go together and move and so on. That's where we find cilia, and an example of that. So that's a unicellular organism. So some organisms have it, some do not. We also have cilia in certain cells of our body, not for movement of the cell itself, but like lining our windpipe, we have cells. This is where your air is when you breathe. This is your cells lining it. So the cells actually have little cilia only on this side. All right, so this side would also have cells, and it'd be actually 3-dimensional, so it'd be around the whole thing. And so you have these little cilia here, and the reason being is that we have mucus that these cells also, some of these cells produce on top of the cilia, and when we breathe in things other than air, like skin cells and bacteria and so on, it collects on the mucus. And guess what? Those cilia wave that mucus up with all that gunk attached to it and brings it back to the back of your throat, and then you swallow it and it goes through down your esophagus and down to your stomach, and it digests it and so on. And so that's an example of cells that have cilia that aren't involved in the movement of the cells. These cells stay put, but they're involved with the function, in this case, of moving the bacteria-laden mucus up to the back of your throat. All right, so another example of that, cilia. And then we have plants also have the cell wall. So a cell wall is an extracellular, meaning it's on the outside, it's the very outside of the cell. We looked at that in the picture of the plant cell. What kinds of things besides plants have it? Prokaryotes, we talked about the bacteria have it, fungi have it, and some unicellular prokaryotes. Those are exceptions to the rule, and they also have some cell walls. The cell wall protects the plant, maintains its shape, and prevents excessive water uptake. So it prevents it from exploding, kind of like when we talked about the contractile vacuoles. The paramecium have contractile vacuoles to help with the explosion, and the plant cell walls help for plants to do that. And plant cell walls are made up of what polysaccharide? You should know this. Cellulose. Embedded in fibers embedded in other polysaccharides and proteins. All right, so therefore, cells rely on a whole bunch of organelles and structures in order to function, and if one of those organelles isn't functioning properly, the whole cell can be damaged. So, for example, a macrophage, this is a cell of your immune system that engulfs foreign particles like bacteria. Its ability to destroy bacteria involves the host cell in coordinating the cytoplasm, cytoskeleton, lysosomes, and the plasma membrane. The lysosomes, remember, have enzymes that can fuse and kill off the bacteria that it engulfs. All right, and that is the cell.